Okay, so again, just a couple highlights from today's class I want to help you out with. If you're having some struggles with this, really might help if you compare these two, mitosis to meiosis. Again, mitotic division here on the bottom, when you take a look at how the chromosomes line up, you can see how each individual chromosome lines up. We're up here when we see this. Now, instead of them being together, we have the actual process of, back in this area, a tetrad lining up. And that tetrad is going to be what's responsible for reducing the chromosome number. And again, the red, let's say in this example, is from mom, blue's from your dad. And so when you make a sperm or an egg cell, looking at these particular cells, for this top one up here, this has much more mom characteristics and genes for that particular chromosome. That one's dad, that one's dad, that one's mom, and vice versa as you work down. So the process here is to reduce the chromosome number and add variation. And that's going to happen in that process here. We reduce it right away in meiosis by having the tetrad line up. We get variation from crossing over during prophase 1. So that formation of the synapse on the tetrad, that's one way to get variation, and how the chromosomes line up on metaphase 1. You know, if they lined up differently, you'd get very different results. So that's what we're referencing when we're talking about mitosis and meiosis. If you notice here, in meiosis, right away, we half the chromosome number from our 46 chromosome for humans to 23 and 23. They parcel out those sister chromatids to get 4 daughter cells with 23 chromosomes that are all different. This process happens, we produce 2 daughter cells that have 46, the full constitution. Okay? Again, knowing the purpose of this is important. Knowing that meiosis produces variation is extremely important. Where does it get that variation? Be a great short answer test question, and it happens because of the way the tetrad lines up during metaphase 1 and crossing over during prophase 1. That gets us the variation. How much variation do we get? That's what your notes talks about there. And what you need to think about is when you look at how many combinations of offspring an individual could produce, you take the formula 2n, where 2 represents the fact that you have the 2 chromosomes, and then the n represents the number of chromosomes. So for humans, for example, we have 2 to the 23rd power. That is based on how the chromosomes line up in prophase 1 of meiosis 1 could get us 2 to the 23rd different combinations of offspring. Well, how many is that? It's going to be staggering, but that produces roughly 838,600,000, I'm running out of space, 608. That's how many different sperm or egg that can be produced by one male, not counting crossing over. Okay, that's a lot of variation. Again, humans or animals, eukaryotes want a lot of variation built in. They get it based on how the tetrad lines up during meiosis 1. But this number here, as large as it is, only accounts for how many different sperm or egg an individual can produce. How about the number of different offsprings? To figure that out, what we need to do is actually calculate the number of sperm or eggs, so that 8,388,608, and I need to square that number. In other words, 2 to the 23rd, oops, that's horrible handwriting, apologize, squared again. What's that number? That is 7,368,744,170,000 different offspring that 2 people can have regardless of crossing over. If you count in crossing over, which is randomness that we can't figure out, it's a much higher number than that. My point is we can, for any organism, figure out the number of possible offsprings they can produce. For example, dogs have a diploid number of 78. Okay, so to figure that out on the test, you would have to take 2 to the 70. That's the diploid number, so you cut 78 in half, right? So you have 2 to the 39th power, and then you'd have to square that to figure out how many different combinations 2 individuals could produce. It's a lot of variation built into this system, and that's intentional. So again, my hope for you to understand is how much variation is possible through meiosis. One of our goals is produce variation. We do that through crossing over and through this process. I would expect on the test for me to give you some organism simpler than this, maybe like a fruit fly, and ask you how many different combinations can be produced. You should be able to calculate that using that formula. I also think it's important to know the stages of meiosis on the test. And at this point, if you're kind of feeling confident, you can turn it off. I'm going to go through a couple of good examples of test questions just to see how well you do. If you want to pay attention, great. If not, you can stop the video at any time. Some test questions you should think about. What is the name of the process that produces sex cells and increases variation in a population? Right? What is that? That's meiosis. Which stage of mitosis or meiosis does crossing over occur? Okay, that's prophase 1. What stage of interphase does DNA double? That'd be a good question to ask on the test. This checkpoint checks for damage to the DNA and makes sure the DNA replicated properly. Which checkpoint would check for that? I would probably have a diagram up there of the tetrad and have it lined up to different things. Something like that, and then have you tell me what the different parts are pointing to. Again, hypothetical question there. That'd be something that'd be more of a matching 3 or 4 options off of. Knowing diploid versus haploid. A question like, a bug has 10 chromosomes in a wing cell. How many different gametes can it produce? Again, if it has 10 in a wing cell, that means it has 5. I do 2 to the 5th power to figure out that answer. I'll let you do the math on that. In males, meiosis is called what? And what does it produce? In males, meiosis is called spermatogenesis and produces 4 active gametes. There'll be cells that you have to understand what stage they're in, whether it's mitotic or meiosis. You want to know about the checkpoints. You get to know where do microtubules of the spindle originate during mitotic division. What is the longest stage of the cell cycle? What is the longest stage of mitosis? What is the shortest stage of mitosis? All questions that could be asked on the test. One more, and then I'm going to close this down. Hopefully I've given you some things to help on. There's not a lot of mark-all that applies, FYI. That's just 2 or 3 I'm catching through as I go through here. There will be some about the scientist, about Pasteur, Pasteur and Francisco Redi. It's more about knowing what they did. Like this theory of spontaneous generation was finally disproven in 1864 by who? Louis Pasteur. So those are the type of questions you'll have on the test. So hopefully this video has helped you. Again, maybe it has, maybe it hasn't. Make sure you're able to calculate that, realize how much variation that you have available to you, and good luck on the test. If you lose your note card, come see me.